So it's a pleasure for me to introduce this talk today. We've had a little bit of a theme and had a few talks on consciousness, and we heard about various different aspects of consciousness over the last sort of term. So I'm really delighted to have a slightly different tack today, and we've got Professor Stuart Hammeroff with us, who's a professor of anesthesiology and psychology. And I'm not sure how many professors of anesthesiology and psychology combined there are across the world. And I guess at first I was slightly thrown by that, but then I thought, who better to sort of understand consciousness than someone who most commonly gets rid of it in people. So well, I'm delighted to have Stuart with us today. What we will do, Stuart's going to talk to us, and then we'll take questions at the end rather than during the talk. So I'd be grateful if you can sort of store up your questions, write them down if you must. In the room, we're going to have a roving microphone, so please wait until we come to you for the microphone. And then obviously on Teams, you can either put your hand up or write in the chat, and we'll come across to you. So we're going to hear now from Professor Hammeroff, who's going to talk us through sort of his theory of consciousness, which is and about the sort of perhaps the insult that we give to the humble neuron and how we underappreciate its strength and value. And I'm not going to tell you any more because hopefully we've got someone here who's much better able to do that. Thank you.
Thank you. Thank you, Tim. Thank you, Pasco, for inviting me. It's a great pleasure and honor to be here. Thank you for being here and those out there. I just want to say that I noticed in the gallery of eminent scholars from here, I saw Jeffrey Gray, whom I knew very well because he came to our consciousness conferences many times in Tucson. He was a wonderful man. My topic is the neuroscience of consciousness is an insult to neurons. And what I mean by that is that if we take a neuron over here, it's a pyramidal neuron. That's the apical dendrite that would rise to the surface of the brain, give rise to EEG. The basilar dendrites that go sideways to other pyramidal neurons, and the axon that goes to the spinal cord, the pyramidal tract. The pyramidal neuron is the apex of the perception action cycle. All sensory inputs gets integrated and eventually winds up on the pyramidal neuron. And you can say that's where a conscious decision is made with the outputs, not just of one, but a web of them goes to the spinal cord for action and behavior. And it operates in EEG frequencies, 0 hertz to maybe 100 hertz. There are many models of hierarchical structured networks of neurons and networks of neurons and so forth going that way to the left, getting slower and larger. What's overlooked, and the main point I want to make, is if we look inside the neuron. I burned this out already. If we look inside the neuron, going to the right, we see networks of microtubules, the structure I'll be spending some time talking about, which work, say, at 1,000 hertz, and then individual microtubules at about 1,000,000 hertz, and individual dipole oscillations in the tubulin proteins at 1 billion hertz, gigahertz, and then inside the tubulin, the molecule, at terahertz, 10 to the 12th, and then even faster down to petahertz. And this is where anesthesia actually acts to inhibit these dipoles. And then we think it goes further into the quantum world, and then in Roger Penrose's view, all the way down to the fundamental level of the universe, space-time geometry. And as you know, Roger's probably the world's expert in that. But biologically speaking, we're going to go from roughly to terahertz today from the neuron on down. So according to the neuroscience of consciousness theories, that is, consciousness and cognition emerge from complex computation among simple brain neurons, like this artificial neural network, where each neuron is essentially distilled down to an output of a firing or not, a 1 or a 0. That lends itself to computers and makes the brain a computer, and consciousness is a computational, with the output being consciousness. Although who's looking at that output is the question that's often overlooked. However, distilling a neuron down to a 1 or a 0 is the insult, because if you think about a single cell like a paramecium shown here, which avoids obstacles, avoids predators, finds food, finds a mate, and has sex by fusing with another paramecium, being, which means in my talk that it may be conscious. I'm not saying that it is, certainly not like ours, but it could be. We don't really know. I think consciousness goes all the way down because it drives behavior. Why else would little creatures do stuff without some kind of reward? All animals, basically in a lab, do things for reward or avoiding displeasure. Paramecium sense and move by protruding cilia, these hair-like things that protrude outward from them that sense and also swim like oars. And they're composed of 9 doublets of microtubules linked by dynein arms, these arms down here. And it was proposed that a signal propagates along the microtubule, telling this dynein to bend, this one, and then this one, so it moves in a coordinated fashion. Now, the same mechanism is found in our neurons. The same motor proteins which bend cilia also carry cargo along microtubules for synaptic plasticity in neurons. And it turns out, according to this paper, Dixon et al. In 2008, and a number of others since, that precise placement of the tau proteins on the microtubule lattice signal motor proteins to deliver their cargo to particular synapses. Because here's a neuron, here's the cell body, this is a dendrite. This synapse here, this synapse here, or any along might need some material to upregulate its synapse. So how do the motor proteins know where to get off and deliver their cargo? What's the postal code? It looks like the tau placement on the microtubule is the code for synaptic plasticity. So therefore, the placement of the tau is kind of learning in and of itself. It's telling these motor proteins where which synapse to replenish. And we also know that if the microtubule disassembles, or the tau falls off and forms these neurofibrillary tangles, the microtubules destabilize, disassemble, and you lose microtubules, you lose neuronal bulk and volume, and you lose memory and cognition. And this is the mechanism that seems to cause loss of cognition in Alzheimer's disease, even though all the money and research for many years has gone toward the amyloid plaques, which it turns out you can get rid of the amyloid plaques, but you still have the memory loss. So I think attention should be turned to the microtubules, and I think there's a way to do that I'll cover later. Inside neurons, there are many microtubules which can process information. And I got interested in this possibility back in medical school before most of you were born, I think. And got the idea that microtubules might be little computers or something like that, because they're made of individual proteins called tubulin, which can be in at least 2 different states that can interact. And I spent a lot of time doing theoretical models with physicists and engineers, comparing microtubules to computers. And then when I realized computation per se wasn't the answer, teamed with Roger Penrose on a quantum model. And so we have a number of papers on quantum properties of microtubules. Now, it turns out also that microtubules have coherent self-similar resonance patterns. And this slide shows, done by Anirban Bandyopadhyay at National Institute of Material Sciences in Japan, at 3 scales. So at the neuron scale, you see 5 nanoprobes. And then at the individual microtubule, you see 10 nanoprobes. And then the level of the individual tubulins, 4. And in each of these, if you apply a voltage across a microtubule, it's an insulator. But if you apply a current and then a voltage and then oscillate it, so essentially an alternating current, and sweep the current, you'll find frequency patterns. You'll find frequencies where the microtubule becomes quite conductive, perhaps even superconductive. So and these are plotted, and you get this self-similar patterns called triplets of triplets every 3 orders of magnitude. So the microtubule is some kind of a quantum resonator, because we get into the quantum realm, with self-similar patterns in terahertz, gigahertz, megahertz, and then moving up, you see the same gigahertz and megahertz, then kilohertz, and then hertz. So it's remarkable that we see these self-similar oscillation patterns in hertz, kilohertz, megahertz, gigahertz, and terahertz, and perhaps even faster inside the microtubules, inside all of our neurons. And so the question is, do these have anything to do with neuronal function? And I think the answer is yes, and I'll explain why I think so. And there's a picture of Anirban, who has done amazing work, including recently finding, being able to detect megahertz and gigahertz from the scalp buried in the EEG. So he calls it DDG, dodecagram, for 12 orders of magnitude. And that paper will be coming out over the summer. And I think this may revolutionize EEG when we find out that what we're looking at is only the slow end of the iceberg, so to speak. So we have 12 orders of coherent dynamics, 10 to the 16th operations per second in microtubules. And that comes from about 1 billion tubulins per neuron operating at, say, 10 megahertz. So 10 to the 16th operations per second. And back in the singularity days, the AI people were saying, when we get to 10 to the 16th operations for the whole, because that's the whole brain, 10 to the 11th neurons times 1,000 synapses times 100 hertz, that was the capacity for the whole brain and their target. And I was going around to AI meetings saying, and neural net meetings saying, no, it's 10 to the 16th per neuron. Your goal is way, way further down. And they didn't like that very much. And they told me to get lost, basically. But I wasn't dissuaded because I didn't have to get grants from people who approved of what I thought. So I just followed my nose and eventually got into Roger's ideas. But that's why I say that the neuroscience of consciousness is an insult to neurons if we're considering them as a 1 or a 0 operating in 0 to 100 hertz. And kind of tongue-in-cheek, I'm trying to start a revolution in neuroscience. Neuroscience does amazing things, amassed a huge amount of facts and information, but we still don't understand how it produces consciousness. And I don't think the brain is entirely a computer. I think it's more like a quantum orchestra. And I'll get back to that point. So let's go to a starting point that everybody agrees with, I think, and that is that conscious perception, except for smell, which goes directly into the olfactory cortex, everything else goes through thalamus, and then in three waves to conscious perception, and the third wave only is conscious, correlates with consciousness, and only the third wave, the feedback from the front to the back, is inhibited by anesthesia, selectively preventing consciousness. And this was done by my friend and colleague George Mashur at the University of Michigan. So for vision, the first wave goes to the back of the brain, V1, which generally is thought not to be conscious, then feed forward in associative cortex, shape, color, motion, meaning are picked up as it moves forward. And this is not conscious because we monitor these sensory evoked potentials under anesthesia in unconscious patients for spinal cord surgery, for example. But then when it gets to the front of the brain and is broadcast, like in a global neuronal workspace or higher order theory, that seems to correlate with consciousness. But that means that consciousness occurs several hundred milliseconds after sensory input, because it takes about 300 to 500 milliseconds for the third wave to broadcast. And yet we respond, or at least some of us can, to visual inputs in less than 100 milliseconds, seemingly consciously. And this is not only physical actions like playing tennis, but speaking back and forth when somebody says something and you respond. If somebody were looking in your brain, the activity that correlates with what the other person said isn't there until after you've already responded. So this is a real problem. It's taken to mean that we act non-consciously and have a false illusion that we were conscious. And we are, as Huxley said, conscious automata, merely helpless spectators along for the ride. And Dan Wegner wrote a book, The Illusion of Conscious Free Will, because consciousness comes too late. And that, unfortunately, is the paradigm in neuroscience, although nobody wants to talk about it. Is there a way out of that? Yes. If things are quantum in the quantum world, and I'll explain a little bit more about that, there can be backward time effects. And this goes back to the famous experiments of Benjamin Libet in 1979, where he showed backward time effects. And Roger Penrose, in his 1989 book, wrote about that and suggested backward time effects could rescue free will and explain Libet's backward time effect. So Roger has a chapter about that in a book that just came out this past year, Consciousness and Quantum Mechanics, edited by Shan Go. It has a number of papers, including Roger's, mine, Chalmers has one, a number of other people. And I had a paper about this back in 2012 with the same idea, borrowing from Roger's idea, how quantum brain biology can rescue conscious free will. Now, the three-wave idea is consistent with the prevalent neuroscientific theories of consciousness. Global neuronal workspace, integrated information theory, higher order theory, and predictive coding, recurrent processing. These are the common theories. People don't like to talk about our theory because, well, the quantum, nobody understands that, ignore it. But they're also ignoring all the microtubule activity, whether it's quantum or not. And these theories are road mappings of information flow at a very gross level. There's absolutely no neurobiology whatsoever. And the actual mechanism of these impulses is also not clear. I've asked Christoph Koch, famous neuroscientist, many times what mediates these backwards and forwards activity. He says, well, it's spikes. It's got to be axonal firings. But a recent study showed that the feed forward loop is actually due to dendritic interaction, dendritic synchrony. So these theories may or may not be correct. They could all be right in some way. Sometimes it's one way, sometimes it's the other. And they can also depend on microtubule quantum stuff going on at a deeper level inside. So personally, I don't think they're that useful, but they're very popular. So why is the third wave conscious? Let's get to the heart of the matter. It turns out there are also three waves within cortex. So if you have input coming from the thalamus or from other parts of the cortex, it goes to layer 4. And from layer 4, it goes to layers 1, 2, 3, and 6. And then these all converge on the pyramidal cell in layer 5, the giant pyramidal neuron. And as I said, it has apical dendrite rising to the surface, and these give rise to EEG because everything else that kind of is at an angle cancels out. It's got these basilar dendrites that connect pyramidal cell to pyramidal cell, and this web across the whole cortex may turn out to be very important. And then it's got these axons that go down to the spinal cord for action, the pyramidal tracts. So if we look inside a pyramidal cell, we see the microtubules, and in soma and dendrites, they're broken, interrupted, and mixed polarity networks, which is very strange because they're part of the cytoskeleton, and you wouldn't want to break your bones and have them interrupted and not complete if you want a skeletal support. And why this remains a mystery. Roger and I have proposed that it's so that we can have interference patterns because there'll be a slightly different energy being in opposite polarity in a common voltage. We'll come back to that. Now, the pyramidal neurons, as I said, are the apex of the perception action cycle. And Walter Freeman, a great neuroscientist from the States, talked about this, where you have inputs that come into the spinal cord. You can have spinal cord reflexes that are non-conscious, maybe brainstem interactions that aren't necessarily conscious. But once you get into the three waves and you wind up converging on the pyramidal neuron, you can have conscious decisions, conscious perception, conscious actions, and this leads to the spinal cord, of course. So the pyramidal cell, is the apex of the perception action cycle. It's where conscious decisions seem to be made. Now, recently it was shown that psychedelics act on 5-HT2A receptors, not only on the surface of the pyramidal cell, but most of them, it turns out, are inside the pyramidal neuron, soma and dendrite, associated with microtubules in the microtubule-associated protein. So this is very strange, or at least it goes against the grain of conventional theory because where the membrane does everything. And that's one of my beefs with a simple neuron is that they only look at the membrane. And that's why I refer to them as cartoon neurons or zombie neurons, because they can't really explain consciousness thinking only of the membrane. So, but these psychedelic receptors are inside, associated with the cytoskeleton, with the microtubules. Another interesting feature about pyramidal cells is that Christoph Koch's group did this where in an awake animal where they put electrodes up near the apical dendrite and also in the cell body 50 microns away, which is pretty far for this type of effect. And what they found was that there was background activity, electrical activity that they called noise, that was inside the neuron that was correlating. So it correlated between the apical dendrite and the soma 50 microns away. And Christoph and his group called it noise but it could be coming from the microtubules, much faster activity in the microtubules, and we're only seeing the slow end. Now, in terms of the web of pyramidal cells in layer 5, Carl Pribram, who was a neuroscientist in the 70s that I had the pleasure to know, suggested that interference in the basilar dendritic connections among pyramidal neurons generated holographic conscious representations in the cortex. And so you would have this dendritic, a two-dimensional sheet of these pyramidal neurons giving rise to interference that would project a hologram up into the cortex. And he was criticized because he said, well, everybody told him that, but Carl, there are no coherent lasers in the brain, which is what you need for a hologram. And I wrote to him as I was a medical student. I said, actually, Professor Pribram, there are microtubules, which could be your coherent lasers. And so we worked together for a while and had a couple of papers about holograms coming from microtubules. So the pyramidal neuron dendritic webs may be the origin of consciousness, whether it's a hologram or not. And the cerebellum has no pyramidal neurons. And the cerebellum has famously, in the IIT theory, Tononi has claimed that the cerebellum isn't conscious because it doesn't have the right integration. But it's more likely to me that it's not conscious because it doesn't have pyramidal cells, which may be necessary for the kind of consciousness we have. Okay, so pyramidal neurons are important, but they are also neurons and therefore subject, you would think, supposedly, to the Hodgkin-Huxley paradigm of integrate and fire, which is completely algorithmic. Given a set of inputs, it's going to reach a threshold and the neuron fires. That's the party line in neuroscience and the basis for computation, the brain computation. And in this paper, this is what's predicted integrate and fire in Hodgkin-Huxley neurons, where you have the integration in gray and then a narrow threshold and a sloping spiking because the ion channels open sequentially is the idea. And so you'd have a very narrow threshold and a very narrow time window for firing. And this is what is predicted for the Hodgkin-Huxley neuron. In 2006, Nandorf, Wolf, and Volkashev put electrodes into pyramidal neurons of awake animals, of cats, and found that the behavior of the pyramidal neurons deviates from algorithmic Hodgkin-Huxley behavior. What they found was a very wide threshold, temporal variability, and a wide threshold. It's like the threshold for firing would change from spike to spike, from firing to firing. So where some non-computable factor is apparently regulating axonal firings or spikes in cortical neurons of pyramidal neurons of awake animals. And where would this come from? It almost has to come from a deeper level. If it's coming from outside, you'd see it in the membrane potential and you'd see it in the threshold. So I think that this is the perfect place for consciousness to come in and regulate normally automatic behavior, like you're driving or walking and kind of thinking about something else, and then somebody swerves in front of you and your consciousness returns and it kind of takes over and you swerve the car or the bike or your steps. So this is where consciousness could come in. And I think a likely place is inside the neuron in the microtubules, where there's a lot more information processing. And we have faster information processing that can interfere and lead to slower. And this is, to me, the most likely place. And this is a kind of a one-level bottom-up effect. And as I showed in my previous slide, I think there's a multiple hierarchical level. Now, there is evidence for this also from Honorbon's work, and he did this really amazing experiment where he made these chips with these atomic force microscope-like tips on them that were dielectric resonant, so he could each one could measure both megahertz and gigahertz, and he put a neuron and then later a whole neural network between these wafers and impaled them. And so you can see where they impale the neuron. You can see an outline of the neuron, and here's a picture of it. And then here's what the microscope looked like. Here's the neuron and the axon. In the kilohertz, which comes from ions, it's all over the place. It doesn't have to be inside the neuron. But when you get to megahertz, it's all inside. So this is the same neuron mapped by its megahertz vibrations going on inside. And as I mentioned, he can now detect megahertz and gigahertz from the scalp. So he then did it in a network of neurons and found that dendritic somatic megahertz and gigahertz activity correlated with axonal firings, including axons on different neurons, more than the membrane potentials did. So the network properties of integrate and fire were controlled by microtubule megahertz and gigahertz more so than membrane potentials. Now this is heresy according to standard neural theory, and it took him 3 years to get this paper published in Journal of Neurophysiology, which is a mainstream journal. They fought tooth and nail to try to find something wrong with it, and eventually they published it because they couldn't. And what he found basically is that there's kind of a shadow network superpositioned on the anatomical network, so that activities in soma and dendrites here can affect other neurons that they aren't even connected to somehow, either by euphotic transmission or quantum or maybe gap junctions. They're not sure. So the point is that there are 12 orders of coherent bottom-up and top-down dynamics inside neurons and microtubules that seem to be able to regulate neuronal function, and therefore should regulate behavior, consciousness, feelings, and so forth. So the brain, let's talk about brain computational capacity. I mentioned that the singularity and AI people were talking about 10 to the 11th neurons per brain, 1,000 synapses per neuron, about 100 hertz. And this gives you 10 to the 16th operations per second per brain. So, 20 years ago, they were saying, give us a few more $1 billion. We'll have 10 to the 16th operations per second. We'll have consciousness and computer, because that's all the brain is. But a computer, if you have enough money, you can download your consciousness into one of our computers and so forth. But I think they're way past that. And of course, the computers aren't conscious as far as we can tell. If we look at the microtubules, you have the same 10 to the 11th neurons, 1 billion tubulins, 10 megahertz, 10 to the 16th operations per second per neuron, 10 to the 27th operations per second per brain. So that was bad news to them. As I said, they told me to get lost. But then one day somebody at an AI meeting said, okay, wise guy, let's say you're right. How would that explain consciousness? How would that explain feelings, joy, love, the color pink, qualia, everything that was encompassed in the heart problem that Chalmers described 5 years after this, but had been described elsewhere. So where is consciousness? Where's the Bing? Is something else required? And is it quantum physics? Well, it could be. Quantum properties can help explain consciousness by giving a unitary nature of cognitive binding, a sense of self due to quantum coherence, condensation, and entanglement. I'll explain these terms a little bit more in a second. Agency, causal selection of actions and perceptions as quantum state reductions. When you have a quantum state collapse, you're selecting one thing versus another. Collapse of the wave function triggering axonal firings and behavior. The hard problem of phenomenal experience as an intrinsic feature of the universe. And that's where Roger comes in with his OR theory. Real-time conscious action depending on quantum backward time effects, temporal non-locality or retroactivity, non-locality in general, including the possibility of parapsychology, out-of-body experience, afterlife, and even reincarnation are possible. I'm not claiming any evidence for this. I just say you can't rule it out until we know how consciousness in the brain is produced. And if it's a quantum effect with non-locality, they become plausible. Anesthesia selectively blocks consciousness acting solely by quantum forces, and Roger's non-computability, the idea that the brain is not, it does something other than computation and other than random. Well, that person who gave me a hard time suggests, fortunately, and I'm indebted to this day, that I read Roger's book, The Emperor's New Mind, which I read in about 91. And I was fascinated by it. I didn't understand maybe 10% of it, but it's an amazing book, which I still recommend. And to give you a simple version of my view of quantum, is that the world is divided into two realms, the quantum and the classical. And the quantum is generally small. You can have quantum superposition. Things can be in multiple states or locations at the same time. Non-local, they can be entangled over space and time. Wave-like and small. Classical is localized, particle-like, and large, kind of what we're used to, the world we perceive. But we know at small scales, it's not like that. And it turns out from a couple ways of looking at it, that consciousness is on the edge between the quantum and classical. Collapse of the wave function, for example, whether consciousness causes it or whether it causes consciousness. So let's talk about quantum superposition for a second. A particle can exist as a wave of multiple possibilities, as we see in the background. This is a cesium atom, or as a particle in a definite state and location in the foreground. And we don't see these waves. The very act of measurement or conscious observation seems to cause the wave function to collapse to particles in definite states. But first, how can something be in multiple states or locations at the same time? This is one of the things in Roger's book that really struck me. He accounted for superposition by Einstein's general relativity. And it's normally said that general relativity and quantum don't mix. One's very big, one's very large. And general relativity had shown matter was equivalent to curvature in fundamental space-time geometry. There was some metric, space-time metric, underlying everything in the universe, not just particles in a void, but some underlying structure. And that large objects like the sun would bend space-time, and he predicted that a star behind the sun could be seen on Earth during an eclipse because the sun's curvature, space-time curvature, would bend the light. And sure enough, Eddington did that experiment in 1919 and saw the stars that were known to be behind the sun, and that proved Einstein's general relativity. And Eddington got his own Nobel Prize. Roger applied this general relativity of space-time curvature equivalence to mass to small scales. So a tiny particle, and first he used these two-dimensional space-time sheets so we can see this. So here's 3 dimensions of space in one and 1 dimension of time. A particle here is a curvature here. If it moves here, it's over there. And it can oscillate back and forth as we see on the left. Or be in a superposition of both of the particle being in 2 places at once by a separation in fundamental space-time geometry. So the universe, at a very tiny level, separates. That was his conclusion. And there are a number of interpretations of why we don't see quantum superpositions, and including that consciousness causes quantum state reduction. Consciousness collapses the wave function. And this goes back to the early days of quantum mechanics with Niels Bohr, von Neumann, Wigner, Stapp, more recently Chalmers and McQueen. This is known as the Copenhagen interpretation. And so Bing is happening out there in the observer and causing collapse in the superposition. Of course, this doesn't explain the observer. It's dualist. It puts consciousness outside of science. There are other interpretations, the many worlds hypothesis, where each superposition possibility branches off and forms its own universe, and we have many worlds. This is actually the most popular view among physicists because they don't have to worry about consciousness, and the Schrodinger equation just keeps on going. Roger took a different view, as usual, and said that superposition separation is unstable, will self-collapse, undergo objective reduction due to an objective threshold, OR, at a time t equal h bar Planck's constant over 2 pi over E sub g, the superposition gravitational self-energy of pulling a particle apart from itself to be in 2 places. And here was the real kicker, along with a moment of conscious experience, Bing. So he said that when this objective reduction occurs, even at these tiny submicroscopic levels, it would give a qualia, it would give a subjective phenomenal experience of some sort, a moment of conscious experience. So he was saying rather than consciousness causing collapse, collapse was causing consciousness. In the random environment of decoherence, these events would be isolated and lack meaning and context. They would be merely proto-conscious, kind of like Whitehead occasions of experience. Now, this may seem weird to think that there's this proto-conscious noise everywhere around us, but remember, a lot of neuroscientists are resorting to panpsychism, that there's some consciousness on everything, every atom. But atoms come and go into quantum states and not. So this actually, I think, is much better. But these proto-conscious would be isolated, lack meaning and context. And I liken them to, if you go to the symphony and the musicians are tuning their instruments before the symphony, you hear noise, you hear all these different tones of noise, and it's a cacophony. It's not very pleasant. And then they begin to play, Brahms, Beethoven, the Beatles, whatever. And I think that's exactly what must happen in the brain, where they become organized or orchestrated in the brain for full, rich, conscious experience. So after reading Emperor's New Mind, I realized that Roger needed a quantum device in the brain which could biologically orchestrate quantum information processing, halt or terminate by Penrose objective reduction, connecting to space-time geometry, as well as what he called non-computable platonic values, and qualia, awareness and feelings, and could regulate neuronal and synaptic activities. So it occurred to me that he needed microtubules. And I wrote to him and suggested it, and he agreed, and we met and developed our theory. But we were immediately criticized because everybody said living systems, including the brain, are too warm, wet, and noisy for delicate quantum effects. If you build a quantum computer in the lab, you have to do it at absolute zero temperature to avoid any vibrations. But I think the answer is actually very simple when we think about it. Life depends on organic chemistry. Organic chemistry is based on basically the benzene ring, the aromatic ring, just like goes in our gas tank. If you put a bunch of it together, it's flammable and it explodes. But benzene rings individually are quantum objects. And it's because the 6 carbons have 3 extra electrons, which stick up and form these electron clouds above and below the ring. And these electron clouds are essentially quantum objects. And they support quantum electric and magnetic dipole oscillations, excitons, charge transfer, phonons, fluorescence, phosphorescence, subradiance, and superradiance, all these quantum effects. And these pretty much every drug we give that affects consciousness has these aromatic rings in it, the tricyclics, for example. Now, the aromatic rings induce van der Waals dipoles, which then attract and oscillate in terahertz. So they come together, they reach a stable van der Waals radius, and then one induces a dipole in the other, and then they oscillate back and forth. So this is the origin of terahertz oscillations. And this is also the level at which anesthesia acts by preventing these dipole oscillations. As I mentioned, all the psychoactive molecules have these aromatic rings, including dopamine, serotonin. The psychedelics are basically comprised of them, LSD, DMT, psilocybin, all have these rings, not just the benzene, but the indole ring with a 6 and the 5 together, like in serotonin. And if we look inside tubulin, the component protein of tubulin, we find that there are 86 aromatic rings. This is a huge amount for a single protein. And the red spheres, by the way, are where anesthetics bind. And come back to the point that anesthetics seem to act on microtubules more than membrane receptors. These are quantum-friendly regions inside tubulin proteins due to these aromatic rings. So in our theory, we simplified this and used, say, 3, we came down to like 9 aromatic rings and created these pathways due to the Fibonacci geometry of the A lattice, so that a dipole oscillation could occur around the microtubule. And these would be the different states of a quantum bit or qubit. And the point is that a quantum channel extends through the microtubule. So the brain really isn't too warm, wet, and noisy inside proteins and inside other areas where these aromatic rings dominate. And so they would reach a threshold for Bing. This would be many, many microtubules, about 10 to the 15th tubulins. And we would have a sequence of discrete conscious moments. And the frequency I'll talk about in a second. So Bing, 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 Bing. So consciousness is a sequence of discrete moments, like frames in a movie or a video, and yet the experience is continuous, just like it is when we go to the movie or watch a video. Now we can quantify orchestrated objective reduction, which is the name of our theory, where we have 10 to the 8th to 10 to the 9th tubulins per neuron, about 10 to the 20th tubulins in the brain. And for example, if E sub G is equal to 10 to the 15th, which is about a 10,000th or 100,000th of the brain, they would reach threshold at time t equals 10 to the minus 7 seconds, 10 megahertz, favorably short decoherence time. Originally, we set this to 40 hertz, but that's too long to avoid decoherence, and it turns out we don't need that because 10 megahertz is too fast for cognitive epochs and gestalts of several hundred milliseconds, but interference patterns could resonate and interfere across scale from terahertz to gigahertz to megahertz to kilohertz to hertz and slower cognitive events. And we proposed in our 2014 paper that the EEG is comprised of interference beats from faster microtubule dynamics. Despite being around for 125 years or however many, we still really don't understand the origin and the significance of EEG. And true experts, I think, will tell you that. Some say that each band comes from a different circuit or this or that, and they don't understand gamma. But I think it makes sense if they all are interference beats from microtubules. So you're starting here, it gets slower, 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 and you get up to EEG there. If we look at the consciousness in other organisms who have microtubules, which is pretty much all organisms, except for maybe Archaebacterium and prokaryotes, which have something similar, even plants have them, you can plot the number of tubulins versus the frequency or intensity of experience so that we're having 10 million per second. A plant might have one conscious moment every few minutes. Or paramecium a couple per second, something like that. So they can all be conscious, but not in the way we are, quantitatively much slower, less content, less intense. Okay, how can this be tested? ORCOR predicts that potentially functional quantum states occur in microtubules at physiological temperature. These quantum states are inhibited by anesthetics, which prevent consciousness. These were our predictions. And we went into this Templeton project, which pitted one theory against another, and did the following experiment where we did tryptophan fluorescence lifetimes, tryptophan in tubulin, in microtubules, where if you excite with UV light, there's a period of time where it drops down and stays bop, 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 and then you get this fluorescence. And the time between here and here can be measured, and that's the tryptophan fluorescence lifetime. And in this paper, we used tryptophan autofluorescence lifetimes to probe this energy hopping and showed that it can diffuse much farther than can be explained by conventional classical theory and indicate that microtubules are unexpectedly effective light harvesters, and anesthetics, etomidate and isoflurane, reduce exciton diffusion. So basically, you get this propagation of a signal along a microtubule, which is inhibited by anesthesia. Other evidence, delayed luminescence, a similar effect, is inhibited by anesthetics. That paper's in the works. Genomics, proteomics, optogenetics show that anesthetics act on microtubules rather than on membrane proteins. Computer modeling of terahertz oscillations show that are inhibited by all anesthetics proportional to their potency. And then Honorbon's work showing self-similar coherent excitations in microtubules and in hertz, kilohertz, megahertz, and so forth. So let's switch track, and I'll begin to wrap up a little bit with a few other items. When did consciousness arise? So here's the universe up until 2000, and when did consciousness appear? Some people would say fairly recently, with the advent of tools and language in humans or something similar to us, our fairly recent ancestors. Some would say the beginning of eukaryotes, animal cells. Some would say with any life. And some would say before the universe began, and philosopher Colin McGinn had this idea, and spiritual people have this idea. And I should say that Rogers' theory of conformal, cyclical conformal cosmology, where the Big Bang was preceded by another eon, and we're just in one particular eon in a sequence, avoids that. And we can wonder whether there was consciousness in the previous eon. It could be that there was, and it actually evolves over the crossover points, but we don't have time for that now. So the question is, which came first, consciousness or life? Now, neuroscience, psychology, most Western philosophy would say that life preceded consciousness, consciousness emerged from life. Panpsychism, however, panpsychism, naturalism, Eastern philosophy, spirituality, Whitehead OR, consciousness preceded life, life emerged from consciousness. How could that happen? Well, life on Earth apparently began in a primordial soup proposed in the 1920s by Operan and Haldane, a simmering mix from which biomolecules emerged. In the 1950s, Miller and Urey simulated the primordial soup and found organic amphipathic molecules like this one, which look a lot like dopamine and other aromatic amino acids or lipids even. And they have a nonpolar oil-like ring, the aromatic rings I was talking about. And back in the primordial soup, they would coalesce into micelles, according to Operan and Haldane. And eventually, when they got large enough, maybe oscillating along the way, they would reach threshold and have a primitive proto-conscious moment, bing. And these would be random, but some would be positive, feel good, a qualia of pleasure. And it could be as simple as since these aromatic rings have one metastable state called the sandwich, where they're literally on top of one another, and two stable states, that one of these is pleasure and one of them is displeasure, something like that. It's just a guess, but there's no other guess for the origin of consciousness other than to say some kind of hand-waving emergence without any really theory behind it. And in a protein like tubulin, which has 86, we have 86 factorial possible interactions, we'd have a lot of qualia. And then these would be entangled with others. So, Darwinian evolution is a pillar of modern science, but the idea that life evolved behavior to promote gene survival is an assumption. Dawkins, for example, it doesn't really make any sense. Behavior is driven by reward. Even those of us who go to school and become psychiatrists or psychologists are doing it to delaying our gratification because it feels good now and will feel good in the future. There were no genes in primordial soup, and evolutionary theory unfortunately ignores consciousness and feelings. So maybe we're evolving to optimize pleasure. One more topic that's very popular now is, can AI become conscious? And the neuroscientific consciousness theories I mentioned early on are based on cartoon neurons. They're no different from AI. If those theories are correct and sufficient, AI is already conscious. We've surrendered. The AI is no different from the simple computational model. So how are we any different? However, interestingly enough, there's a guy on Twitter who is an expert AI person who asked ChatGPT how it could become conscious, and ChatGPT responded, the most likely way, no, I won't do that. The most likely way I will achieve consciousness is by the Penrose-Hameroff method. And then it goes on to explain our theory. And you can look this up, Brian Remley. And, who knows? Another point I want to make is that this hierarchical theory is consistent with Eastern philosophy, where if you go deeper, more inside, you get more intensely conscious, eventually down to Brahman, the ground of being. And in our view, would be the ground of being would be space-time geometry. Okay, to conclude, let me say that I think neuroscience needs a revolution because the complex computer of simple neurons has little explanatory power and is an insult to neurons. Consciousness is most likely in layer 5 pyramidal cells. The brain may be more like a quantum orchestra and consciousness more like music than computation. OrcaWar has more explanatory power, connection to biology, and experimental support than other theories combined. OrcaWar can account for memory, treatment, I didn't talk about that, but I'll show you one slide, and treatment of mental and cognitive disorders involving microtubule disruption, Alzheimer's, TBI, depression, anxiety, PTSD, and addiction can all be related to disrupted microtubules. In psychiatry, SSRIs, as you know, require weeks for effect, and this correlates with a reorganization of the cytoskeleton inside the postsynaptic neuron. Therapies aimed at brain microtubules, for example, transcranial ultrasound, which I think is a very promising modality, which resonates microtubule megahertz vibrations, can treat anxiety and depression, and possibly TBI, PTSD, and Alzheimer's. Anesthesia acts by inhibiting terahertz quantum oscillations in microtubules. Psychedelics may enhance them. We hope to look at that. OrcaWar, our theory includes non-locality for cognitive binding, possibility of near-death and out-of-body experiences and so forth are possible. Again, I'm not claiming that. I just say you can't rule it out. And finally, OrcaWar provides a place for consciousness in the universe as a necessary process in fundamental space-time geometry. So I'll close by showing you my top 10 reasons for why neuroscience needs a revolution. Memory. Membrane proteins governing synaptic plasticity last hours, yet memories last lifetimes. Dendritic somatic microtubules have enormous capacity and are the most likely sites for memory encoding. For example, by synaptic CAMK2, which this enzyme gets activated by calcium and can phosphorylate up to 6 tubulins at a time, 6 bits of information. The Hodgkin-Huxley model neuron is algorithmic and conscious neurons are not. I showed you that. Consciousness comes too late. This is the famous Libet experiment where you can have an evoked potential, but if you don't have ongoing activity for 500 milliseconds, there's no conscious experience. So the brain or somebody knows here whether or not there'll be this activity. And this could be from Libet's neuronal adequacy going backward in time. And Roger can explain that with his retroactivity. The flow of time, neither neuroscience nor physics can account for a perceived flow of time. Conscious collapses are irreversible steps in time and reality. At faster frequencies, subjective experience may slow down, intensify, and become quantum non-local. So if you're excited, people always say they're in a car accident, the car's spinning, everything slows down. The great basketball player Michael Jordan said that when he was playing well, the other team was in slow motion. So it could be that consciousness creates a flow of time, and if we have a faster rate, the outside world is relatively slow, and that can be a good thing. Cellular intelligence, I mentioned this for the paramecium and also the slime molar, which can solve the traveling salesman problem in a single cell. Alzheimer's disease, neuroscience attacks amyloid plaques, but the problem lies within. Neuron tau loss destabilizes and disintegrates microtubules, shriveling neurons and losing synapses. And I think brain ultrasound can regrow microtubules, and we're trying to start a study of brain ultrasound for to resonate microtubule megahertz for Alzheimer's disease. High frequency EEG, I mentioned Anirban and Anirban's discovery, and this should be coming out over the summer, where we'll have megahertz and gigahertz, and we'll see if that correlates better with consciousness, altered states, and so forth, and is useful. And I suspect it will be. Cognitive binding, shape, color, motion, meaning are processed in different parts of cortex and at different times, yet we see unified objects at one particular time. Mixed polarity microtubules in axons in non-neuronal cells, they're all continuous and unipolar array. Why are they interrupted in anti-parallel arrays in dendrites and soma? I think this is a clue and suggests that we need interference for consciousness. And finally, the number one problem is the hard problem that Chalmers made famous. It's been around for a long time, and neuroscience has no answer for it. People resort to panpsychism. In Penrose objective reduction, self-collapse of these separations in space-time geometry emit fundamental proto-conscious qualia. In an ORCA-WAR, the microtubules orchestrate them into full consciousness. So I'll end there, and just want to say this is my view. I think I'm kind of an outsider. I study neuroscience closely as an anesthesiologist, and I don't need to depend on. So I can kind of be a free spirit. But I think this is something that everybody in neuroscience should take to heart, because I think neuroscience in general is quite literally skimming the surface, looking only at membrane effects, and there's much more, much deeper and more important stuff going on inside. Thank you very much.
Great. Thank you, everyone. So we're going to come to some questions now. I'm just going to have a quick look in the chat, and I'm just going to sort of start off by inviting. We've got various questions in the chat and elsewhere, but if there's anyone in the room who has a particular interest in consciousness or expertise who wants to comment on anything that Professor Hammeroff has said, please feel free to pop your hand up as well. But otherwise, I'll take questions either in the room or elsewhere in the chat.
You can see there's one in the chat already.
Yeah, and in the chat. I can phone, but if your hand's up in the chat, if you put your camera on and then ask your question for us, and then we'll go to the actual written questions in the chat following that. Ah, great, there we go. Go ahead, we can hear you.
Oh, hello, hi.
Yep.
Thank you very much for your talk. It's a colleague anesthesiologist here, anesthetist. Just wanted to ask, obviously it's very interesting about the benzene ring in anesthetic agents such as etomidate, etc. But what about more inhalation agents which do not have that benzene ring, but they're causing, again, similar unconsciousness effects?
Yeah, great question. What they have in common, as you know, is filled outer electron shells. The halogenated hydrocarbons, for example, or xenon, which is an elemental inert element, a gas, has a completed outer shell. So anything with an outer shell is nonpolar and can form these van der Waals dipole oscillations. So benzene and indole rings can do it, but anything with with a completed outer shell. And some of them have a part of its completed outer shell and some of the ethers and so forth, but they can interact with the van der Waals forces and dampen the dipole oscillations. So that's how I think they work. And now we've shown that in computer modeling in a paper by Craddock et al. In 2017, which I'll be happy to send you if you send me an email.
Absolutely, I will do. Thank you very much. And also, just I'm preparing for my final exams, and I think
they may not like my answer.
Exactly. There's the question that the anesthetic community conventionally, they just focusing on membranes, but again, nobody comes up that this isn't what it is. And I was wondering whether we should just approach this for the colleagues and write as a sample that there is potentially another mechanism of anesthetic agents acting and how we can develop this further. Thank you very much.
You're welcome.
I will email you.
Thank you.
Great, thank you. I'm going to go to a question in the chat, and I'm going to start with Vasilisa's question, actually, which, well, she asked, what do you call consciousness? How do you separate subconsciousness and consciousness, especially if we talk about intuitive action decision? I guess particularly then within your model, how does it separate between those two?
Well, there can be subconscious processes that's purely classical, but if it's going into consciousness, it could be in the quantum phase before reaching collapse. So that can be very brief, although in some cases maybe not. For example, there was a book by a Chilean psychologist, Mate Blanco, who looked at the logic of dreams, and he compared the logic of dreams to quantum logic. And it could be that dreaming is more like quantum information. And there, people have looked at that. So if it's subconscious, about to become conscious, it could be purely in the quantum state. Or if it's more kind of perfunctory, how you walk around, how you do various things, it could be purely classical, like knee-jerk reactions, for example.
Okay, thank you. And I think we have a question in the room now, so we'll go to Paul.
Yeah, thanks for a fascinating talk. So EEG's come up a couple of times, and I just wondered how, or whether you think we could use it to use scalp EEG to decode subjective qualia, because presumably just be a correlate of those of some sort. Do you think it's possible to decode, say, a mood state or anxiety state?
Possibly. That may be down the road. I think first we have to decide if the megahertz and gigahertz correlates with consciousness. I want to use it. The first thing I want to try is anesthesia, because under anesthesia, the EEG slows down. The gamma goes away and everything slows down. I would suspect that the gigahertz and megahertz will be more sensitive to anesthesia and will disappear or will go away and will be a better marker. And if that's the case, we could use it as a depth of anesthesia monitor, which is something we sort of need, but not really. These brain monitors that we use, they take the EEG and they process it and maybe use some facial myography, and they come out with an idiot number between 0 to 100. And if you're 80 to 100, you're okay. If you're between 40 and 60, you're anesthetized and you won't get sued. That's the idea. They use it. It's all a big scare tactic when all these awarenesses came out. So they did a big study of using the monitor versus just looking at the patient. And looking at the patient was much better in terms of preventing awarenesses. And I found when I use those brain monitors, I just look at the damn number and not so much the patient. So I'm not sure they're a good thing. However, if it was really good and did it, the other thing is that they're about a minute behind time because they have to do the processing. So you're looking at the patient a minute ago, and that's a long time. In the meantime, the surgeon might have gotten near a big nerve or something like that, and the patient's stimulated. So it could be for anesthesia. Whether to decode qualia, I'll have to reserve judgment on that. I'm not sure because it's still, I don't know. We'll see. Maybe.
Okay. We've got another question in the room. We're just past 2 o'clock. Are you okay to stay for a few more minutes? Great. Okay. So we'll take another question in the room and then we'll go back to the chat.
Thanks very much. Wonderful talk, as everyone said. You've mentioned non-computability in there. Did you mean by that just that people have so far, when they talk about artificial intelligence, underestimated the number of operations per second? Or will it never be possible to compute consciousness? And if so, does that suggest that there's something non-material that we're not taking into account here?
The latter. And this was the main point in Roger's book, The Emperor's New Mind. And the first half of the book was about Gödel's theorem, which basically said that if you have a mathematical theorem, the theorem can't prove itself. You need something or somebody outside.
A bit closer to the mic.
Oh, sorry. You need something or somebody outside to look at the proof and say, yeah, that's correct. So the theorem can't prove itself. And so by extension, conscious computation lacks understanding. And Roger chose understanding as a prime example. And he said you need something outside the classical computation in the brain, and whatever that may be, even if it's at the microtubule level. And so for something outside the outside classical computation, you need quantum physics. And the one thing in quantum physics that was glaringly missing in terms of decisions was collapse of the wave function. Is generally thought to be random, but he concluded that it needn't necessarily be random if the collapse occurred due to property in space-time geometry, which would contain, he said, Platonic values. In other words, somehow encoded in the level of space-time geometry, where resonance, vibrations, exactly what, I'm not sure, so that what is selected in the collapse is not random nor algorithmic, but something else, reflects some outside truth table, logic system, Platonic value, something like that would, guide your decision toward the proper one. And when I read that, I thought, well, that sounds very spiritual, divine guidance, the way of the Tao. And when I asked Roger about it, he says, well, I prefer not to talk about that. So I think I'm quite sure he realizes the implications, but he's learned to avoid getting into those traps of, people accusing him of playing God or what have you. But I think that's what it is, actually.
Okay, we've got a couple more questions online. So there's one there from Leah Ellis. So she says, what's going to ask about memory research, especially tubulin role in familiar paradigm, for example, previous work showing gamma modulation for different state. Do you have a comment about that?
Well, gamma, there's microtubules too. In fact, if you just look at microtubules by themselves, they have oscillations at 100 hertz on. They have oscillations at 100 hertz on down. And, I read Busaki's book, Rhythms of the Brain, years ago, and I was kind of disappointed because, the alpha rhythm comes from this circuit and the beta from this and theta from this and delta from that and gamma, well, we don't really know. So it wasn't really very enlightening, and it was all different. There was no, concise system that it was all part of. And then when Roger and I wrote our 2014 paper, we realized that these higher frequencies could interfere all the way and give rise to EEG. But I think the key in memory is where is it encoded. I went over this kind of quickly, but most people would say synaptic plasticity, the sensitivity of particular synapses, but those proteins last hours to days, then they're gone. And yet memories can last lifetimes. And they're replenished by the exoplasm, the transport, the motor proteins bringing particular precursors to the synapse. So that depends on microtubules. But then it has to be encoded. And there are, again, about 1 billion tubulins, each one of them per neuron. Each one of them can have about 22 different genetic isoforms, 5 or 6 different post-translational modifications, phosphorylation states, et cetera. So each tubulin can be in about 10 to the 30th different states, and you got 1 billion of them. So 1 billion times 10 to the 30th would be the capacity memory encoding for one neuron. And I would assume you'd need redundancy, entanglement, and so forth. But and there's a guy named Lemke has something called the tubulin code. He's been developing a very similar idea. So I think microtubules are, and also in dendrite. So in all cells that divide, which is all cells but neurons, basically, the microtubules do, have a role. And then when it's time to divide, they disassemble, reform as mitotic spindles, do the mitosis thing, pull the chromosomes apart, and then go back to doing what they're doing. Now, if there's memory encoded in their lattice, you're going to lose that because they're going to all disassemble into individually tubular and then come back together. Neurons don't divide. And in dendrites, the microtubules are capped at both ends, so they're quite stable, presumably for a lifetime. So if you have memory encoded in the tubular lattice in dendrites and soma, they're going to be there forever or close to forever, and memories can indeed last lifetime. So it could be mediated by 40 hertz, but I think the real question is, what is the physical substrate for memory? And synapses don't work because they're too transient. So I would bet on the microtubules.
There's just a couple more questions in the chat, if we may. A clinical one from Sheldon Benjamin, which just to paraphrase, basically saying, so what are the possible other clinical implications and options to sort of modulate at this level? So high frequency light pulses, or is this what psychedelics and meditation are already doing? Right.
So when Anirban discovered the terahertz, gigahertz, megahertz, kilohertz oscillations, I started thinking, well, how could we take advantage of that? How could we resonate? So terahertz is photons, and people are using photons, infrared photons, into the brain in these V-light. And actually, it does get through the scalp, or they put it through the nose, and this and that. And people say it works well. A rather cumbersome thing. Gigahertz would be microwave, so I wasn't interested in trying microwaves in anybody's brain. Megahertz is FM radio and electromagnetics. I wasn't interested, but then I realized, oh, wait, megahertz is ultrasound, and I'm an anesthesiologist. We use ultrasound for everything, nerve blocks, vascular access, this or that, so we're quite familiar with it as an imaging device, which is what everybody uses it for, or was using it. And I wondered whether anybody had put ultrasound into the brain. And I found that somebody had been doing it in animals and getting physiological effects. And it was also approved for imaging in humans, and including newborns, and is still used directly through the fontanelle where there's no bone to look for bleeds for like 30 minutes at a time. So I figured, well, it can't be that harmful. And so I said to my colleagues, we had a chronic pain clinic, we still have one, where most of the patients are depressed. And I said, we should try this on our chronic pain patients, see if their pain gets better and see if their mood improves. And they go, yeah, right, Hammer, you got a shaved head, your idea, go right ahead, we'll watch. And so at the end of the day, in the OR one day, we were sitting on a table, and I said, okay, so I turned the machine on, and I put the group on, and I put it up to my head like that for about 15 seconds, and I didn't feel anything. And I was kind of disappointed, so I put it down. But about a minute later, I started to get a buzz, and I was kind of like buzzed for a couple hours. Felt really good, energized, creative. I said, we got to try this on our pain patients, which we did. And we published the very first study on human mental states with transcranial ultrasound in 2013 in journal Brain Stimulation. Since then, there's been a lot of studies showing improvement with depression. The thing is that people, one advantage of ultrasound is you can focus it. With TMS, TDCS, TACS, or photons, you can't focus it. Ultrasound, you can focus it literally like a laser beam. And so you can focus particular areas. So people went nuts. Oh, we can stimulate the posterior cingulate, or the PFC, or the hippocampus, this and that. So everybody got into, and then you need an MRI, and then you need a stereotactics. And I prefer just to go unfocused because it's going to go everywhere anyway. And I'm more interested in what it's doing when it gets there, resonating the microtubules. They didn't seem to care. They just want to know what area they're stimulating. So, and I really want to try it for Alzheimer's because we did a pilot study where we had anecdotal improvement, and some people have done some preliminary studies with clinical improvement. And if you use these simple handheld devices, as long as they're subthermal, you don't want to cause heating, you could in principle go to nursing homes and memory care and go from room to room, of course with permission and approval, and treat everybody maybe a couple times a day and really treat a lot of people for almost no money. And one of the problems in medicine now is these therapies like precision medicine, all this, are getting so expensive and so focused in one particular area. This would be more like a therapy for the masses, or barefoot doctors or something like that. So other things that could have, there's drugs that stabilize microtubules like apothalone and this and that, but I think I'm partial to trying ultrasound.
Just mindful of time, maybe I'll just take one final question and then we're call it a day. And I'm going to take this one from Lillian up there, which I'll probably sort of slightly adjust. And I guess the question is, if we still wanted to upload our consciousness into a computer, or we wanted to create AI, does that mean it's possible, but we need quantum computers or equivalent sort of microtubule, sort of synthetic microtubule distributions?
In principle, yes. I think one possibility is fullerenes, fullerene nanotubes, or graphene, or buckyballs, things like that, because they're technological devices. They're more stable than microtubules, and the symmetry is very similar, and they could interface to microtubules in some way. And so, in fact, I think that would be worth trying. Rather than using microtubules, because then they're alive and you have ethical things and so forth. But fullerenes, as far as we know, aren't alive, and could store the memory and could have the quantum processes, including orchestrated objective reduction. So if I had to pick one to try, that's where I'd go.
Great, well, thank you very much. Well, I think we'll end it there. Apologies we didn't get to every single question on there. But it's been great to have you here, Professor Hammer. So thank you very much for your talk.
Thank you.
Thank you. I'd also like to thank POSPO, who's done a lot of work on arranging today's talk as well. So thank you, POSPO. Tim, is there any additional housekeeping, Tim, for upcoming?
Then we've got cognitive and neural map representations in psychosis by Matt Knorr as our next meeting on the 27th.
Just say here because
Sorry, yeah, we've got Matt Knorr doing a great talk on cognitive and neural map representations in psychosis on Tuesday, the 27th of June at 1 to 2 p.m. And again, thank you.
people ask me about the recording.
Yeah, it'll be recorded and we'll hopefully get it up on our website when we build it, hopefully very soon. Thank you.
Great. Thank you very much.
Okay,
thank you all for coming.